northern tip of France. And so we're doing this for a very small area. But we have the capability to run these predictions over a much larger scale. And by linking it to the environmental parameters, the hairball, or the metabolite structure, we can actually say what the carbon consumption or reduction of each of these grid squares will be. And so we've done that here, slightly more 1970s now style cartoon. And what we see here is changes in the ability of the ecosystem over time. We see that September, October, November, December, the community to produce or consume carbon dioxide. So we're predicting for the Western English Channel the ability of that channel to generate in every grid square carbon dioxide or to consume carbon dioxide. So whether a particular location becomes a carbon source or a carbon sink based on the microbial population dynamics in that system. That is pretty awesome. Because what that enables us to do is now start linking this into global climate prediction change. Because one of the fundamental drivers of global climate prediction change is release of methane and carbon dioxide from specific environments. If we can now start forecasting this, we can actually forecast this by 6 days at the moment. We're hoping to explore that and see if we can go further, because 6 days doesn't give us a lot of warning. But if we can start forecasting this by weeks or months or decades, then it will make life a lot easier. This is weird, I know. But this is the sound of the microbial community as it's changing over time. This is my last slide. This is microbial music. Yeah, this is something I did for fun. Done by Peter Larson at Argonne National Labs. But this shows you that you can actually translate the microbial community into music. So we have another one. These are the rare microbial communities. So I'll shut up for a second. Now this has got to go to number one, seriously. So these are the cyanobacteria, the Vibrialis, the Bacillialis, Pseudomonalis that bloom up from being very rare to being very abundant. And by translating this into music, and we're using a very complicated method, so the environmental parameters are actually the chords, and the timpani or the arpeggiation of that chord is the community structure. I think that's brilliant. Anyway, I won't hammer home because you can also make it into a requiem. So yeah, that's brilliant. But anyway, thank you very much. There's lots of people I can thank, but I'll put their names up. Thank you.
Thanks, Jack. Interesting.
Sure. So what we are doing is we're using 60S and 80S. So we've done the profiling of the 60S for the first 5,000 samples. That gives you the bacteria. 80S will give us some analysis of the fungal communities in the soil and the fungal communities in the marine environment, which are actually quite abundant. And also tell us about the phytoplankton and the zooplankton in the liquid system and the protists that exist in the soil system. Now, we are trying to work on an intergenic spacer set of primers with various groups, including Jed and Rob, who will be in tomorrow, to try and understand if we can use the ITS region between the 16S and 23S and the 18S and 25S to actually improve that community profile. Remember, the 16S, the 18S, the ITS studies are very, they're profiling. They're only helping us to see how different each sample is. The real data, the real detail, and what we've seen there, the importance comes from actually looking at the metagenomics or the metatranscriptomics or the proteomics. Once we have that information, we can start to analyze and integrate and interrogate that community structure. I'll let you be
My name is Fan Chu from Chinese Academy of Sciences. Actually, I have some collaboration with Rob Knight and Red and about the Arctic tundra soil. My question is about soil sampling. As you know, because if you collect 1,000 samples, some sites very good, but some sites very bad. So after soil sampling, how do you translate soil sample to the lab? I mean, do you think the microbial community will change during the transportation or not? And another question is, we know microbial community may change with season, I mean temporal change. So if you collect 1,000 samples, I don't think you can collect at the same time, right? So do you think, how do you compare? If the sample is not at the same time, how do you compare the microbial community?
So we're comparing the I'll answer the last question first and the first question last. But we're comparing the microbial community based on its environmental parameters in which it finds itself. So there is actually an ongoing initiative to do, we'll call it a World Ocean Sampling Day. So the major oceanic sampling centres are coming together to take samples on one day of the actual summer solstice, so June 21st in 2014, to actually take all the samples on one day and analyse them all at the same time, using the same methods as the EMP standard protocols. So that would actually fix that one point. However, it doesn't really matter if they're taken in different time points. If you have a time series, because you can understand the successional development of that community. If they're not taken in a time series, but they're taken geographically disparate, such as the Global Ocean Survey or Tara Oceans, what you do actually have a significant problem. I agree. Your problem stems from being able to relate each site to each other site. So if you have the environmental characteristics, you can actually use those environmental characteristics to define that community at that time in that place. So it may not be necessary to have a time series to actually define how that microbial community looks at that particular location. It's not ideal, but it may help.
I understand. Thank you.
So, okay, is there any questions?
Oh, one more. So,
okay. That's only one question?
Yes, just a devil's advocate position for a second. So we know soils are incredibly diverse, a lot more than water, okay? And so is it possible that by choosing a standard method, you actually make for a greater bias and convergence than if you figured out the best method for each sort of individual soil sample that gave you equal efficiency? So what this slide may indicate, it gives you that unexpected result of lower alpha diversity in soil than stream water. Is that not just because of a standard extraction protocol from very different types of soil?
No. The stream water was heavily contaminated with soil that leached out of the banks of the river. And so there was a lot of soil particulate material in the stream water. And so what you actually see when you compare the microbial community here to here is that a lot of the OTUs up in the soil community map to their OTUs in the freshwater stream. That was unexpected. But it explains why stream water may have actually a higher alpha diversity because it has the stream water diversity plus some of the soil microbial diversity actually including there. I agree, though, wholeheartedly that the issue of DNA extraction, and Tom Delmont may talk about that a little bit in his talk, actually has a fundamental role to play in defining how we actually explore microbiology. But to use the devil's advocate to finish up with one last statement, sometimes you've just got to suck it and see and pick one particular method and go with it. Because otherwise you won't have comparability and we will have to start differentiating and branching. That's for the future of society.
But if you knew that you were extracting DNA from 90% of the cells, like continuously, regardless of what you use, then you would actually have potentially better comparability than a standard method where you didn't know how many cells you actually extracted from, sure, depending on the soil.
As long as you got the right 90%, and one wasn't so heavily biased towards removing one particular phylum in that 10% that didn't get picked up. So yeah, ideally, we would have something which was so robust it picked up everything that we have to make a decision somewhere. So that's what this conference is all about. Trying to uncover and define. We should consider talking about it.
That's Jack's very interesting talk. And next speaker is Bernie Perro, and